Proteins could be classified into two categories for generation of new proteins during evolution: convergence versus divergence. Any two proteins with not sharing for their primary sequences, but similar 3D structures. Those proteins considered as convergent evolution for their structures, 3D structures. Instead, any two proteins with sharing primary structures but different functions and structures could be considered as divergent evolution. Third group in this class will be Leucine-rich motif. More than 60 proteins had been found containing this type of motif, such as receptor cell adhesion molecules, RNA splicing, and DNA repair proteins. Ribonuclease inhibitor, which is 456 residue long. There is some 20-30 repeating units. There are two types: 29 amino acid type A, 28 amino acid type B. Two types are repeating 15 times alternatively, plus other the sequences as well at the termini of chain. Repeating unit shares a certain conserved residues in type A and B, mostly by hydrophobic residues in two secondary structures plus in loop. In real three-dimensional structures, conserved hydrophobic residues get together, making hydrophobic core. In contrast, the other side of parallel betas are exposed to the solvent, which is different from the barrel type. Also, this is important in terms of application because we can construct plausible structure models with Leucine-rich motif. For the second group, open twisted beta structures, it has some common active sites, highly conserved. As you see here, in terms of topology, there is some topologically switching point, which just represents reverse direction point for the two motif, which just make very big crevice. So for these proteins, just active sites are positioned this crevice. It's called topological switching point. Further, this category open twisted alpha beta proteins, it's very varied for length of beta and number of beta and actual arrangement of betas. Which is different from the team data, which is very strict in number of beta and alpha, typically 8 numbers. But open twisted tissues here, they are a lot varied. The aeroshell tRNA synthase has this kind of open twisted alpha slash beta domain, expanding the active site, detailing binding to the substrate. Another type, carboxypeptidase, much complex pattern determined by William Lipscomb in Harvard, but still active sites is conserved in switching point, expanding the active site. This is another protein belonging to this category, arabinose binding protein. Much complex, but still it's just active site is positioned in the switching point. Details the substrate binding regions. So anyway, could be summarized this way. Many proteins consist of 2 alpha slash beta open sheet domains. Active site is in crevice between 2 domains. 2 domains are arranged in a way that the carboxy edge of beta should extend towards active site.